Shreyasi, and today we'll be talking about complementary genes. So, we already know about Mendel's laws, and as Mendel said that any character has two alleles for it, two contrasting traits. Okay. And we also know that if the genes for both the traits are present together, only one of them show. One of them masks the effect of the other. But the gene is still there in the next generation. The gene which is getting masked of the recessive allele might get expressed. Now, we already know about the dominant recessive interaction. But complementary genes, this concept comes from the concept of epistasis, a phenomenon of epistasis. What is it? In dominant recessive interaction, at a single locus, the dominant allele is somehow masking the recessive one. But in epistasis, two different genes in two different locus interact, and one of them suppress the expression of the other. So let's say in one locus we have gene A. There's another gene B. Gene A has its dominant form, which is like capital A. It has its recessive form, which is small a. Gene B again has its dominant form, which is capital B. It has its recessive form, which is small b. Now, if sometimes it happens that gene A is masking the expression of gene B, okay, and there are many ways this interaction can happen. Complementary gene. So here the gene which does gets expressed is called the epistatic gene, and the one which is getting repressed or suppressed is called the hypostatic gene. Complementary genes is a case of recessive epistasis. Okay. Now we'll just get a little more into it, a little more detail. Exactly happens in complementary genes? Just like I showed you, there are two separate genes. It can be in its dominant or recessive form. Each of them give rise to a similar trait. So the point here is that two genes give rise to same character or property rather independently. Okay, but if you have two sets of genes on different loci, maybe on separate chromosomes, and if present they'll give rise to the same property independently. But if present in their dominant forms, that is, the two genes I'm talking about, if both of them have their dominant alleles present together, they give rise to a new trait. Let's just recapitulate once again. Complementary genes is the phenomenon when we have two separate genes on two separate chromosomes. Both have their dominant and recessive alleles. They separately, independently give rise to a same property, same or mantles have the dominant allele present together, it will give rise to a completely new character. Okay, it will become easier with the next example I am about to give you. But make a note of this, please. Now, this phenomenon has been observed in a plant called sweet pea, or the scientific name is Lanthyrus odoratus. What has been observed in sweet pea? There are two genes. Okay, one is gene C, the other is gene P. Okay, obviously the dominant here is capital C, recessive capital C, and the recessive is small c. Here also dominant is capital P, recessive is small p. What happens in this case is, in normal times, gene C and gene P are responsible for a white colored flower. Okay, but if in a particular plant capital C is present at one loci and the capital P is present in the other ones, that is C and P, both exist in their dominant form, you get a purple flower, which is not possible in any other combination. So let's see. So, if you have capital C and P, you will get a purple flower, and any other combination, capital C, small p, okay, or small c, small p, or small c, capital P, any combination where capital C and capital P are not present together, the flower is going to be white. Okay? This bit is clear. If it is clear, then we will move on to the checkerboard. Make note. So, if we move on to the cross now, I'll just rub this as well. In the parent generation, I am crossing a plant with capital C, small p, with a plant with small c, capital P. From what I have told you before, since in any of these plants, capital C and capital P are not present together, both are going to be, okay, can we have a different color? Yeah. They're both going to be white. What are the gametes? So what we get from this? You are going to get capital. These are the gametes you will get, right? From here, right? Next, in the F1 or the first filial generation, what's the organism that you get? What's the genotype you will get? You will get capital C, small c, capital P, small p. And what's going to be the color? You see capital C, capital P are present together. This particular organism is going to be purple. Clear? Next. In the F2 generation, we are going to cross the two individuals from the F1. So when we have capital C, small c, capital P, and the rest, I'll rub it and make a checkerboard. So I hope you've followed till now. In that too, our organisms were capital P, small p, into capital C, small c, capital P, small p. Okay? Checkerboard. So what are going to be the gametes here? The male gametes are going to be capital C, capital P, capital C, small p, small c, capital P, small c, small p. Same again. Okay. Now. Let's first write down the genotypes and then we'll determine the phenotype. Yeah, I hope you're up to me till now. Now we'll determine what color each of these individuals are going to be. So just do it with me, okay? What did I say? Whenever the capital C and capital P genes will be present together, the organism is going to be purple. Other cases, it will be white. So let's see. Capital C, capital P present. So this is purple. Capital C, again, purple. Again, purple. Again, purple. Here also, purple. See, over here, you have the capital C gene, but you don't have the capital B gene. So, this one's going to be white. Again, this is going to be purple. Again, it's going to be white. Okay, white we should write with this color. Again, purple. Okay, now just think clearly. What is the dihybrid ratio of pentyl? And whenever I say the word dihybrid ratio, remember we are always talking about the phenotypic ratio. So, original dihybrid ratio, you know, from Mendel, we all know is 9:3:3:1. Look what has happened here. the number of purple plants is to the number of white plants, or rather flowers. What is happening? If you count, there's 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 purple is to 7 white flowers. So you see how drastically the two ratios have changed, just because these two genes located on two separate chromosomes may be interacted and gave rise to a completely different phenotype. Okay? So this is your complementary gene or recessive epistasis ratio. It's important for you to know. And if you have got this covered, copied it, whatever, I'm going to tell you a little about why this exactly happens, and after that we can wrap up, but just know this well, okay? And that ratio is very important. Okay, now, what exactly does a gene do? A gene, as you know, is a piece of DNA, just giving rise to a particular RNA, which will code for a particular protein. Now, both this gene C and gene P give rise to particular enzymes. And you know, we all know from what you have studied earlier or now, enzymes are very specific, okay? They are very specific for which substrate they are going to act on. But it's also true that some biomolecules, they resemble each other a lot. So cross reactions are also observed at times, and this is what happens here a little. Okay. What's important is, say the substrate is S. Okay, what is it? It's a substrate. The gene C, and so you have the gene C, which gives rise to gene C enzyme, this particular enzyme, which from the substrate produces a particular compound called a chromogen. Okay. And what's important is, gene P, as I told, it encodes for an oxidase enzyme. And what happens here is, when a chromogen is formed and capital P gene encodes for an oxidase, the gene P oxidase acts and produces anthocyanins. And do we know what anthocyanins are? Anthocyanins are those colorful pigments which gives color to your flower petals. So, gene C, gene P are there producing two different enzymes, doing their work or whatever. But if present together, you see both the enzymes are going to be there, and gene C is going to code for chromatin. It will be produced. And if gene P oxidase gets a chromatin, it will produce anthocyanins, which would not have been possible in the small p condition. Also, if the small c was just there, this chromogen would not have been formed, and that is why we are getting these anthocyanins, these pigments, only if both the enzymes are present, both the genes are in their dominant condition, and you get the anthocyanins, and obviously you get a colored petal, you get a purple flower. Okay, and so this is just a short overview about what's exactly happening there, why it is suddenly like two genes located elsewhere are suddenly interacting and showing this dominant recessive effect. Why? So this is a short overview on that. But what you have to remember here in this entire is, first of all, you need to know what is epistasis. Okay, that two genes located on different loci interacting. In complementary genes, which we discussed today, you need to know that it's a case of recessive epistasis. Okay, that here we have two genes showing characters, same or similar character independently, but together in the dominant form is giving rise to a new character. A new character is a new phenotype, and so the dihybrid ratio, the 9:3:3:1, is getting altered significantly, and it's giving you a ratio of 9:7. With that, I wrap up.